All right, so this is an overview. Now, what I do want to make sure you understand is that there's DNA replication, and that's in preparation to do mitosis. Okay? Then you also have protein synthesis, making proteins. And there's two major events that happen there. One is something called transcription. And there's another thing that's going to be translation. This happens in the nucleus. This happens in the nucleus. This happens in the cytoplasm, specifically the ribosome. DNA replication, transcription, translation, probably the three most difficult things to deal with. Please do not get the three things mixed up. A lot of students do. They get mixed up with DNA replication and transcription, and they have nothing to do with each other, although this deals with DNA polymerase, this deals with RNA polymerase. You could see where they came up with these weird things, okay, to confuse people, okay? I finished with DNA replication. Obviously, I did finish with mitosis. Let me just give you a brief synopsis of protein synthesis, and tomorrow we'll go into the details of transcription and translation, but today I just want to give you an overview by saying let's make a brownie. We'll get there. I'll show you what I mean, okay? It took me a long time to come up with that analogy, but it works. First, what happens is the DNA is going to unzip. Already you're thinking DNA replication. Don't. Okay? It's done differently. Okay? But we still have to unzip it. Transcription is then going to take place. And it's going to make a copy of that protein, whatever we need to do. The recipe for that protein. When it makes it, it's going to make it as mRNA, because we can't take that DNA outside the nucleus. But we can make a copy of a certain page in that encyclopedia set and bring it out. That's called mRNA. The mRNA goes out in the cytoplasm and attaches to the ribosome, and that's where we're going to have translation take place. Translation is where tRNA is going to come onto the ribosome where the mRNA is. Follow me so far? Okay. And a tRNA carries an amino acid that's going to be attached to the next amino acid, creating this protein. A long chain of amino acids is called a protein. Okay? And then it gets carried to the Golgi apparatus, which is going to get packaged. Okay? And it goes through this whole thing over here. Now, this is, you can put your pencils down, it'll all be on the recording stuff. This is basically my idea of what's happening. Let's say we want to make a protein. We want to make a brownie. The brownie is going to be my protein. And I want to make the best brownie on earth. So I need to go to the library, to the nucleus. And they have a big encyclopedia set that has 46 volumes in this encyclopedia set of all the recipes that are the best out of this world, recipes in the whole world. Okay? I want to get that recipe for that brownie. I'm not allowed to take that book outside the library. It's a reference book. Okay? So I don't want to make a copy of the whole recipe, of the whole volume, the 46 volumes. Not going to happen. I just want to make a copy of those 3 pages in that recipe book. Does that make sense? Of that brownie. So I want to make that protein. So what am I going to do? I'm going to open up, unzip the specific book. There's 46 volumes. It's brownies under volume 2. chromosome number 2. On page 23, it's on the short arm of the chromosome, whatever. Okay? I open it up, unzip it, and I'm going to take that still in the library, and I'm going to make a copy of those 3 pages on the Xerox machine. That's called transcription. Then I'm going to take the book, close it. I didn't disturb it. I didn't rip out any pages or write on it. Close it and put it back on the shelf, and I got myself 3 pages of this mRNA, the copy, by way of transcription. It took place in the library, it took place in the nucleus. Now, also in the nucleus, I have these pages here. And you know what? There's some stuff on there I just don't want. There's a few lines from the previous recipe that I really don't care about. I'm going to cut that out. There's a picture of Julia Childs. I really don't care about that. So I'm going to cut that out over there. I'm going to splice things and put it all together so that it's now on 2 pages and I got the recipe that I wanted. Okay? I'm going to take that recipe, mRNA now, a copy of the recipe that was on the DNA. I'm going to take that outside the nucleus. That's what the mRNA is going to do. Take it outside into the cytoplasm. I'm going to follow endoplasmic reticulum metro, okay, the roadway to go to my house, which happens to be the kitchen of the ribosome. I really thought about every part of this, okay? So I go to the ribosome, and now I got the recipe there, and I got the bowl there. I don't have the ingredients, and I have to follow the recipe to a T. If it says that I got to put water in the bowl first, and then put the egg, and then put the brownie mix, and then put in water, I have to do that particular order. But I don't have the ingredients in my kitchen, my ribosome. This is where I need to call up my mother, my father, okay? They are the tRNAs. And they're going to be carrying the amino acid to put into the bowl. And I have to call them in a particular order. It says milk comes first. So I got to call my mother who has the milk. She's tRNA. She's carrying the amino acid of milk. She's driving up to my driveway over there. She comes into the house, to the ribosome kitchen, and she takes the milk and pours it into the bowl. She leaves the milk with me, and she goes back into her car, and she leaves without the amino acid. Okay? Now, meanwhile, the next thing it says is eggs. Dad's got the eggs. So I call up his number. His number, like my mother, I didn't say that, but my mother is going to have a certain phone call. I got to call her. And then it's a code because she has the milk. He has the eggs. I know who to call up. That's something we call the genetic code. Okay? I call up my father who has the eggs. He comes over, drives up to the driveway, comes into the kitchen, cracks the eggs, and puts it in there. Notice that this bowl is getting longer and longer with all these ingredients, all these amino acids. He leaves now without the amino acid. My sister comes in with the brownie mix. She's the next person that comes in. She pours in the brownie mix. And she leaves without the amino acid. And my brother comes in with the water, pours in the water, and so forth. You're making this chain of amino acids, which is going to be my protein, which is the brownie. I'm not finished, right? Now, all the ingredients are there. I put it into the oven. Ding! It's all done. I take it out. Okay? The brownie is made. The protein is made in the ribosome. And I can't eat it right away like that. Like I would want to, just stuff my face with it. But I gotta package it. And I gotta cut it up into squares. Some of them I'm gonna eat right away. No doubt. Okay? Some of them I'm gonna put in Tupperware, close it, and leave it for a later use. I'm packaging it. This is the Golgi apparatus, the Golgi complex. All right? The brownie's already made. I'm packaging it into what I think is going to be useful in this way. I'm going to take some of those squares and put saran wrap around them, and I'm going to give them to other neighbors, other cells. Okay? That's basically what's happening here. The whole thing, we're in the ribosome where mom is bringing in the milk and dad is bringing in the eggs and sister's bringing in the brownies. That whole process, that's called translation. So the transcription is making a copy of the recipe in the nucleus. The translation is actually adding all the amino acids together in the ribosomes and making a long chain, making this brownie or protein. Does that make sense? Okay?
So transcription only occurs in the nucleus?
Correct. All right. I just want to do one more. I just want to show you one video showing that whole thing. Not the brownie, but I'll show you what I have over here, and it's putting it together. Okay. So that's how this all happens. What I just explained to you. This is the nucleus up here. Here's where the mRNA is being made, which is a copy of that. The instructions to make that brownie, the recipe, happens in the nucleus. Goes out here. The ribosome comes together over here, so there's a close-up of the ribosome. tRNA, which has an amino acid, comes in over here, takes the amino acid, attaches it to the bowl, so to say, and then this amino acid, the transfer RNA leaves without the amino acid. Okay.
With computer animation, we can enter the cell to view this remarkable system at work. After entering the heart of the cell, we see the tightly wound strands of DNA, storehouses for the instructions necessary to build every protein in an organism. In a process known as transcription, a molecular machine first unwinds a section of the DNA helix to expose the genetic instructions needed to assemble a specific protein molecule. Another machine then copies these instructions to form a molecule known as messenger RNA. When transcription is complete, the slender RNA strand carries the genetic information through the nuclear pore complex, the gatekeeper for traffic in and out of the cell nucleus. The messenger RNA strand is directed to a two-part molecular factory called a ribosome. After attaching itself securely, the process of translation begins. Inside the ribosome, a molecular assembly line builds a specifically sequenced chain of amino acids. These amino acids are transported from other parts of the cell and then linked into chains often hundreds of units long. Their sequential arrangement determines the type of protein manufactured. When the chain is finished, it is moved from the ribosome to a barrel-shaped machine that helps fold it into the precise shape critical to its function. After the chain is folded into a protein, it is then released and shepherded by another molecular machine to the exact location where it is needed.
So again, what we have over here is, remember the three processes that we have. We have something that's DNA replication, and that's what we did yesterday, where I showed you the, where we have to duplicate our DNA in preparation of becoming or to go through mitosis, through its division. So after DNA replication, then you go through mitosis. And mitosis is another big process, but it's not so bad, okay? Then I kind of introduced you to making a protein. We'll say protein synthesis, or i.e. making our brownie, right? Because there is tons of proteins that your body has and needs, all right? So much, you know, a lot of desserts you want. You just want to make one particular dessert, the brownie. In order to do this, we have two big processes, and that's what we're going to talk about today. One is going to be transcription. And again, transcription, when we refer back to my brownie analogy, you can't take the encyclopedia set out of the library. You can't take that DNA out of the nucleus. So you're going to just look at one recipe in that encyclopedia set, on that one page, in that one volume, and you're going to make a copy of it using a Xerox machine, close the book, put it back on there, and then you have what we call mRNA. And that mRNA is a copy of that recipe that's going to leave the nucleus, go out in the cytoplasm, go through the endoplasmic reticulum, the roadway to your house, which is the ribosome. And when you get to the ribosome, you're going to do a process called translation. You're going to translate that recipe into an actual brownie. And that happens on the ribosome with the help of mom and dad and all the other transfer RNAs. Okay? So there's three big processes that we're doing. And please don't get mixed up with the enzymes and the parts. It's very easy to do that. DNA replication and transcription, we zip it. We unzip the DNA. But this we use helicase to unzip it. This one we're going to talk about RNA polymerase. Yes, you do have DNA polymerase, but it's located in here, not over here. Very easy to get mixed up with these things. Just be well aware of that, okay? All right, so let's talk about transcription now. All right, making that copy of that recipe from the encyclopedia set. This happens in the nucleus, okay? And I do have a video of this too, I'll show you too. So transcription, we have something called RNA polymerase. So this is not going to be DNA polymerase that's used to make the copy of the, or to duplicate DNA. This is now RNA polymerase. It's going to attach to a certain part of that DNA, the one that you will want to make that certain protein. I want to go through that whole encyclopedia set, and I want to make, and that encyclopedia set is all for desserts. I just want to make that brownie. There's many brownies there, but I want to make this particular one with the walnuts and the raisins and the extra chocolate chips and all that stuff. That's what the one I want to make. There's other ones in there. You can make it with nutmeg and I don't know, prunes or whatever. I don't want to make that one. I want to make this one. So how do I know? Well, I looked in the contents or whatever. I know exactly what volume to go, and I know exactly what page to go. This is what we call the DNA promoter. I'm going to look for that to start. It usually has TA, or TA. We call it a Tata box. Okay, that usually is the beginning of some sort of protein that needs to be made. Okay, so it's a certain sort of sequence of DNA bases. Now the DNA is going to unwind. Okay, and RNase polymerase is going to do that. Not helicase. Don't get mixed up with that. I'm trying to emphasize it even on the video here. Okay, don't get mixed up with those two. So this is the RNA polymerase is going to unwind and open, or quote unquote unzip. We're going to open up the book in the library, okay? And we are then going to start making a certain strand of RNA. a copy. We're going to take that onto the Xerox machine and make a copy of that page. Where there's a C on the DNA, we're going to put a G there. Where there's an A, we're going to put, since this is RNA, a U, uracil. Remember, there is no thiamine in RNA, so it gets replaced with a uracil. Then the RNA, once it's done, it's going to rewind it back, put it back. We're going to close the encyclopedia book and put it back on its shelf undisturbed. We're not ripping out pages, we're not writing in it, it's undisturbed in that way. Now I have a copy of that recipe. Now keep in mind, it's 3 pages, God bless you, but there's stuff on there I really don't care about. Like I said, there could be something from like the recipe that was before the brownie is also, some parts of it is on that page too. Right, the ending of another one, or the beginning of another one, or, you know, picture of Julia Childs. I don't care about that. I just want the meat and throw out the fat and gristle. So we have to change, we have to splice things up also, okay? There's also something called a terminator. The terminator is really where, in the page of the book, when you see the start of another recipe, you know that's the end of the recipe that you want to make a copy of. So there's a terminator end to it, okay? So this yellow thing up here is the RNA polymerase. It's going to go to a certain part of the DNA, and it's going to open it up, and it's going to start making the mRNA, a copy of the DNA. And this whole thing starts being made out. That's the mRNA, messenger RNA. Again, it's just showing you here and making you appreciate which one. Now, there's 2, it opens here. It knows exactly to make a copy on this side, not this side. Because it'll make something totally different over here, because these are different. This C goes with that G up there, so you're going to make a G here. You'll understand it. It'll make a totally different mRNA if you use this side. How does it know? I don't know. We're still trying to figure out things. All right, but it does pick the right side to do that. You know, if the rest, it's like when you open. I'm just thinking as we go along. If we open up the book, how do you know to Xerox the left side of the page, not the right side? Well, it looks like the brownie's on here. It looks like there's ice cream over here. How do I know that? I don't know. I just glance at it. I didn't read it, but I could see it's talking about ice cream. I need a brownie. It picks on the other side. Okay. But if we had it where only one mRNA is being made at one time, and yet we got tons, thousands of proteins that need to be made, you can't do it one at a time. So what happens here, this is the DNA down here, but mRNA, we can make multiple different mRNAs at the same time. It's kind of like the recipe book. If there's 46 volumes over there, and I'm going to be taking a book and I'm going to make a copy, and there's a long line to say, well, yes, but he's using that book. I want to use another book. And there's a few other Xerox machines. So what's happening here is that those 46 volumes, we could have more than one person make copies at the same time. It happens faster. We could utilize it better. Okay? I thought out this thing pretty well. Every nook and cranny of it. Okay? Alright, so this pre-mRNA. So this mRNA is made, but there's some other stuff we have to get rid of, right? We got pictures of Julia Childs and things like that. Now, this is a weird thing, and just accept it. Okay? Did I tell you there's this list that I'm taking with me to my grave? I didn't talk about that yet? Alright, I'll be talking about this list quite often. There's a list that I have that, as I'm looking at things through life, it doesn't make sense. And it's because certain people have decided to call it that, and people are just accepting it. But it's so confusing. And I'll show you, this is one of the things. Okay? So I know I'm writing this stuff down on the list, and when I go up to heaven, if I'm chosen to go up there, God's going to say, oh, who's that Gamaro guy? Oh my God, you know. He's going to see me up there and say, look, I got a list of things here. Who's the one that came up with this? Because I got a bone to pick with him. He's over there and over there. He'll give me all the places. All right? Because I'm going to ask them why they chose to do this. Okay? So the exons, okay, are the things we're keeping. The introns are garbage. Wouldn't you think it's the opposite? Okay? So I don't know what to tell you. You just got to accept it. And then when I die, I'll be thinking of you. Okay? I also have a list that goes to God too. Alright? Where it's not about a person that did this, it's about God. And believe me, the platypus is the #1 thing I'm going to go up there with. You get a beaver and put a beak on it, make it lay leathery eggs like an alligator, make it swim. Doesn't make sense. But those are questions that I have. So there's a couple lists that are going to go into my pocket, and I'll deal with all that upstairs. Okay? If I'm chosen. He might just say, you know what, your time's not here yet. Go aggravate down there a little bit more, okay, before you aggravate me. Okay? Alright, so that's what I'm saying. Exons are the good stuff, the introns are the bad stuff. Okay? It's not a typo. It's just what it is. Okay? And then we have this splicing going on. The best way I could explain it is through the picture. So what's going to happen here is making an mRNA. This is the pre-mRNA right after xeroxing it during transcription. All the dark areas in here are the introns. They're going to get spliced out. The picture of Julia Childs, the recipe from the previous recipe, we're going to cut all that out. We just want the one of the brownie that we're going to do. Then we got to figure out, well, you know what? There was a picture up there, but then we're going to take that the words over here. We're going to bring it up here. We're doing a lot of splicing. And that's what's happening. Maybe it's better for, and that's what I'm doing actually making these videos in too, because I'm putting like other videos inside. You'll see what I'm talking about when you watch them. But you're taking this, you know, you put an A and then B is better over here, you put the C and D over here, and you do all this splicing. And that's what alternative splicing is all about. Okay, it makes it a little bit better for the recipe to be utilized to make that protein. Okay? So, if you think about it, again, don't answer it, just think about it at this point, and I'll ask everybody. But the sequence of DNA bases is right there. The sequence of bases in a strand of mRNA transcribed from this sequence of bases in DNA would be what? Just take a look at it, give it a few moments, don't block the answers. All right? Who thinks A, B, C, D, E? Yeah, D is the answer. Okay? The reason why is because we're making mRNA. So you're using uracil. You wouldn't use thiamine in there. Thiamine would not be found in RNA at all. There's 3 forms of RNA: mRNA, rRNA, and tRNA. There is no thiamine in there. You got to use uracil. Okay?
Transcription is the process by which RNA is assembled from a DNA template. The 2 strands of the DNA double helix are held together by bonds between complementary base pairs. DNA encodes genetic information in the sequence of bases along one strand. The portion of the DNA molecule that is a single gene or coding region is bounded by termination and promoter sites. A molecule of RNA polymerase binds to the promoter site. It moves along the DNA, separating the 2 strands of the double helix. Each now unpaired base will bind to a nucleotide in the vicinity which has the appropriate complementary base. In the synthesis of RNA, uracil is the nucleotide complementary to adenine. This process ceases when RNA polymerase reaches the termination site. The DNA strands bind to one another once again as the new RNA molecule moves away. This RNA is a copy or transcript of the message contained in the gene.
So let's do translation. A little bit more detail. So now the mRNA, the copy of the recipe that you just Xeroxed, is leaving the nucleus, leaving the library, and you're going to drive to, using an endoplasmic reticulum, you're going to drive to your kitchen in your house. And this is where you're going to translate the code, the recipe for the brownie into the actual brownie. All right, so the ribosome is going to translate that sequence, and the ribosome, like I said, is it doesn't have a membrane there. It's actually two subunits kind of clammed together. There's a 40S subunit and a 60S subunit, and they just clam together. Okay, in the middle is where the mRNA is going to be sandwiched in there. Okay, there's three sites that happen on there. So you have this ribosome over here, and on the ribosome we have three sites: A, P, and E. And they kind of, I don't even have to give you a mnemonic for that one, right? Okay. They have a reason for the names of them. I forgot. Pedestal. They have some Latin names for it. So, and they have these three sites. What happens here, we have a tRNA that'll kind of look like this. And then there's going to be an amino acid that's over here. Okay? The mRNA is going to be riding along here. What happens here is the tRNA is going to go to the A site first, come to the P site, drop off its amino acid, go to the E site just before it breaks off without the amino acid. And I got a video to show you this and some pictures, and we'll go into the details of it. It's an assembly line, and that's the way you got to see it as. Okay? But we have a genetic code. How do I know that my sister is carrying the brownie mix? How do I know that my mother is carrying the milk? Let alone I need to know what their phone number is. There's some kind of code, and we call this the genetic code. Okay? We have a base triplet, and that's a sequence of 3 DNA nucleotides that stands for an amino acid. So the 3 DNA nucleotides are in the nucleus. So we're seeing the original one. We call them the base triplets. Things here are always happening by threes. Then we got a codon and anticodon. The codon is located on the mRNA. For instance, the mRNA will have something like A, U, G, C, C, U, A, G. This is the, again, this is the M. Let me do it in black so you can see this. So this is the mRNA. And every 3 bases over here, this is what we call a codon. It's on the mRNA. On the tRNA, we have something called an anticodon. Okay? Before I show you what this is, we have a bunch of different things on here. There's by threes. And each one of those is going to code for a certain amino acid. In the chemistry, you know that there's 20 different amino acids. They code for a certain amino acid. In my analogy, the amino acids is brownie mix, milk, eggs. Each one of those is amino acid. How do I know to call mom who's got the milk? Because this tells me to call mom. Because mom has a code over here that will correspond. This will be UAC. You see it? The U goes with the A goes with the U, the C goes with the G. If I had amino acid, a transfer RNA that have AAA, that would not fit over here. This anticodon would not go here. You see? It's got to fit. The codon will match its corresponding anticodon. With these codons, there's a total of 64 combinations we can do. You won't have to memorize it, but you will have to know how to come up with this. Very easy to do, and I'll show you it. They will code for the 20 amino acids. There are certain codons that will code for 4 or 5 of the same amino acids. For instance, I could call my mother who's got milk, but I could also call Aunt Tilly. She's also got milk too, a different phone number. I thought about everything. Okay? Some of them, however, won't code for an amino acid. It'll code for some instruction. To start, to stop, something like that. So we say it's a stop codon if you have these. It signals it to say, we're all done. My sister was, and you usually will see that code in a recipe by saying, enjoy. Right? When you see enjoy, you know there's not going to be anything more after that. Okay? A start codon might be number 1. It tells you that's what the first thing you're going to do. Usually the start codon is AUG. And it also usually codes for methionine. I'm not going to ask you to memorize all the different amino acids, like biochemistry. But you should recognize what an amino acid is by just a 3-letter symbol, and I'll show you. This is the genetic code. Very easy to manage. It looks busy, but very easy. If I give you, for instance, the codon AUG, how you come up with that is easy. On the left-hand side, you get the first base, A. Up there is the second codon, U. So you're going to know that AUG is going to be in this category here. Don't jump the gun, just look over here. The third base is G, which is over here. So you're going to go A, U, G, and you follow that, and it's right there. Okay? This will code for methionine. MET is methionine. You won't have to know the real word, but you should be able to tell me what amino acid is, at least the abbreviation. Okay? So if I give you a codon of UUG, take a moment, don't yell out what the answer is, but looking at that genetic code, what abbreviation would you be for what amino acid? Just take a moment to, and I'll... All right, what do you think? What is it? I don't know, I can't hear it, so I'm awful. But here, we'll do it. All right, I'm sure you got the right answer because it all sounds in unison. So it'll be U, U, G. So this will be Lucine. Is that what you said? Right? L-E-U. And I would just expect you to know what L, just write down L-E-U. Because you're going to have something very similar to this on your practical or your lecture exam tomorrow. All right? Let's do another one just to make sure. Let's say you had something like G-A-A. Give it a moment. All right, what do you think? What is it? Yeah, everybody sounds the same. All right, let's just do it. G-A-A. So G-A, so you know it's going to be in this area here, G-A-A, which is here. So it's glucine. Okay? Sound good? Yep. Let me do one more because people are still making those crunched up faces. All right, let's say we want to do C, C, C. What do you think? Pro, right? Let's just do it. C, C, so you know it's going to be in this area here. C, it's going to be proline. All right, which you would just write down P-R-O. Okay? Not too bad? Pretty easy. Come on, it's easier than Sudoku. All right? It is. But looking at it for the first time, WTF? Yeah, okay. It's not that big. How they came up with this, again, people who have no social life. Okay? No idea how they came up with it. But it works, and that's the codes. Okay? And like I said, that's the code to make sure that you know that mom has the milk and dad has the eggs. You know the exact phone number, you know who has it. Okay? Alright, so here's the translation steps. Now that you understand what an anticodon and a codon are. The mRNA is going to attach to the small portion of the ribosome. All right, so this is where the black mRNA is going to attach there. Then what we have here, now we're putting it all together, we have the tRNA is going to correspond, has the anticodon, and is going to correspond with the first one over here at the A site. Okay? It comes on here, it moves over to the P site over here. And what happens here is that this, it's hard, I can't really, but this is going to go onto here, moves over to the P site. The amino acid comes off at this point here, and then this moves to the E site without the amino acid, and then it's going to come off UAC with the amino acid. It's going to stay here. Does that make sense? Okay? Then what happens is we have another amino acid that comes in. It's going to go, I'm not another amino acid, but another transfer RNA. But it's got to go with CCU. That's our next one. This moves along over here. Right? So it's going to be, it's got to move, the mRNA is going to move this way, allowing the CCU to go into that space. Now you have another amino acid, or another transfer RNA that has a different amino acid here. So that's another amino acid. And this is going to have a different code here. It'll have GGA. See how I'm doing it? This corresponds to CCU. That's going to come over here, and it's going to break off its amino acid, and it's going to be actually the bond. There's a bond in here. This is a covalent bond that it binds to. It has a special name, a peptide bond. A peptide bond is a covalent bond that connects amino acids together. And covalent bonds are very strong. So this is going to go there as a second one. Actually, would it help you if I say that this is number 1, 1, and this will be like number 2, 2. And then the transfer RNA is going to come off of there. CGGA. The transfer RNA is coming off there without the amino acid. What's happening here is that we're building a chain of amino acids in a particular order. Has to be that order. You have to put in the eggs before you put in the brownie mix. You got to put the milk in before you put the eggs. It has to, otherwise it's not going to taste the same. Okay? And this thing becomes very long. Okay? And they keep on doing this until you get to a stop codon, and then it stops. Alright? So this is just showing you mRNA is this. They have codons on it. The tRNA kind of looks like this. This is the anticodon with the amino acid at the top over here. This is all made up of RNA, that kind of stuff. Yeah, RNA stuff. Okay? So again, here's what I kind of drew for you. There's the mRNA in the process. That's our ribosome. The APE site, I kind of did it backwards, but APC, I'm sorry, APE. These are the codons. This is the anticodon, and you're making this long chain of amino acids that's going to be our protein, our brownie. Okay? China's doing the same thing. Transfer RNA comes in over here. There's the amino acid, and then it breaks off without the amino acid. Mom is not leaving with amino acids, okay, without the eggs or milk or whichever. All right, so you can look at it this way, step by step. They got numbers showing you as amino acids, so you can see it. Okay, and same way over here.
Does the 3' end go into the ribosome first? Or is it the 5'?
I think it's the 3'.
Okay.
I think it's the 3'.
Which side does that go into the A side or the E side first?
A.
A side.
A side. Okay? And again, if we just had one ribosome making one protein and that's it, not good. So what's happening is we have all these blue things over here are the ribosomes. But this mRNA can go through many different ribosomes, and we can make proteins, not just one, we can make a ton of them. It's like 3 chefs in the kitchen. The eggs could be utilized in different things. Does that make sense? Okay? So that we could actually increase the rate of making proteins by having the ribosomes making more and more of this. Okay? And again, this is the whole cell. Transcription occurs in the nucleus. That's making the Xerox copy in the library. The actual making the protein itself is going to be in the cytoplasm, and that's in our kitchen, which is outside the library. So if you think about it, if a tRNA anticodon sequence is UAC, what would the codon be for an mRNA? Remember, don't call out the answer. Who says A, B, C, D, E, okay? Which I kind of did for you over here. Hopefully that's not the reason why you did it. Okay. All right, so let me show you the video translation since there's a lot of characters involved with this.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the amino acyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid, the first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.